Okay, during the last lecture we talked about the three from the four basic tissues. We talked about the epithelium, the connective, and we talked about the muscular tissue. اليوم, today we are going to talk about the nervous tissue. Our objective to know the structural and functional feature and how to distinguish of this basic tissue from the other tissues. And we are going to talk about the cells and the component of the nervous tissue. Nervous tissue is a basic tissue specialized for communication. So the one that responsible for the communication between the different parts of the body is the nervous tissue. And he can do this by the wires. And these wires, which is part from the nervous system, is called the axons. And also the nervous system, beside the function of the communication, also provide secretion for indirect support of communication. Attributes of the nervous system. The first one, irritability, which means the capacity to detect and respond to the changes in the local environment. And by mean the local environment, the external and the internal environment, and usually this done by the communication. The other function, which is the conductivity, the capacity to transmit these responses from the organs toward the nervous system or vice versa, from the nervous system toward the organs. The nervous system anatomically is composed of two portions. We have the peripheral nervous system and we have the central nervous system. The central nervous system, which is consists of the brain and the spinal cord, whereas the peripheral nervous, which is consist of the nerves. And these nerves, we have two types of nerves. We have nerves that emerge from the base of the brain, which we call the cranial nerve, and mostly supply the structures within the head. We call them the cranial nerve, while the other nerve that emerge from the spinal cord are the spinal nerve. As a function, they consist of motor or sensory component. Okay, so in the skin, we have transducers, sensory organs that transmit the information toward the brain. We call it the sensory, whereas the one that carry the information from the brain and provide orders, for example, to take an action, contraction, for example, is called the motor. The neuron is the functional unit of the nervous system. By functional units, I mean the smallest unit can perform a function. We call it the functional unit. And for the nervous system, the functional unit is the neuron. But in our nervous system, not only we have the neuron, we have other supportive tissue. And this supportive tissue, we call it neuroglia. And these neuroglia provide mechanical and metabolic support of the neuron. Usually we don't have connective tissue inside the nervous system. Instead, we have cells, and these cells, which are the neuroglial cells. Usually the neurons consist of three basic components. Consist of the cell body, which another name is the soma or perikaryon, and we have the axons, one axon, and we have the dendrites. Usually the classification of the neuron could be classified based on the function. We have sensory, which is the efferent, or motor, which is efferent, and we have neuron that connect between the sensory and the motor, and these we call the connector or the integrator. Other classification, the number based in the number of the processes. Each neuron has only one axon, but it could have one dendrite, multiple dendrite, or sometimes they don't have any dendrite. So, as I said, based on the function, we have efferent and interneuron. The sensory receive sensory stimuli, motor control the effector organs and the exocrine and endocrine function, and the interneuron, which is the form the link. As I said, based in the number of the processes, we could have multipolar, bipolar, pseudo-unipolar, or unipolar. For this one here, the bipolar, you can see here we have one axons and we have one dendrites. This one's called bipolar. But if it has one axons and multiple dendrites, we call it the multipolar. If we have one axon without dendrites, we call this type is unipolar and only is found in the embryonic tissue. We could not find it in the adult tissue. In this one here, we can see that the axon and dendrite they fuse before they enter the cell body. This one appears as unipolar, but it consists of dendrite and axon, so it's actually not unipolar, so we call it pseudo-unipolar. And this mostly we found it in the dorsal root ganglia in the area of the spinal cord. See, all of them have one axons, but the difference in the number of dendrites. No dendrites, unipolar, one dendrites, bipolar, multiple dendrites, we have the multipolar. We start about the neurons. As I said, this is the functional unit of the nervous system, which is the smallest unit that can perform a function. As I said, it consists of three basic components. We have the cell body, which is the perikaryon or the soma. We have the axons, and we have the dendrites. We can see here, this is the cell body, and here we have the dendrites, and here we have one axons, and we can see the axon extend toward the effector organ, for example here for the muscle cells. The cell body usually is spherical, large, pale staining, centrally located nucleus with a prominent nucleolus. So the cell body, the shape is spherical, very large. One main component is pale staining, and the nucleus are centrally located with a prominent nucleolus. Their cytoplasm contain a very highly well-developed rough endoplasmic reticulum and Golgi apparatus. As we know, rough endoplasmic reticulum is major function is with the protein synthesis, and as we know that neurotransmitters are proteins, so we need a lot of protein synthesis inside these cells. So these cells have a very well developed rough endoplasmic reticulum, and this very well developed rough endoplasmic reticulum with ribosomes is called the Nissl body or the Nissl substance. Also we have neurofilaments, and also we have microtubules. Usually these neurofilaments and microtubules are concerned with the cytoskeleton of the cells, also with the transport inside the cells, and usually the microtubules in their size are larger than neurofilament. Also with aging, there will be a collection of undigested material inside the neuronal cell body, which we call the lipofuscin. So this is the neuronal cell body. We can see these granules, which is the Nissl substance. This is the nucleus, pale staining with a prominent nucleolus. All of these substance indicative for rough endoplasmic reticulum. See again here, this is the perikaryon. This is the area of the nucleus here, prominent nucleolus, and all what we can see here, this is the rough endoplasmic reticulum, which is called the Nissl substance or the Nissl body. Again, all of these are neurons, and we can see this is the nucleus, nucleolus, and we can see here the processes. As I said in the beginning, each neuron only contains one axons. As I said, with aging, there's a collection of this material inside. All of these are the lipofuscin. As I said, only we can see it with aging neurons. The second structure we have in the neuron is the axons. Any structure that conducts information away from the cell body, we call it the axons, and usually for each neuron only we have one axons. The beginning of the axon is called the axon hillock, which is short pyramidal shape area. This region is free for ribosomes, rough endoplasmic, and myelin sheath. The plasma membrane of the axon is called exolemma, whereas the cytoplasm is called the exoplasm. As I said, the exoplasm contains abundant amount from the microtubules and neurofilaments, which concern with the cytoskeleton and the transport inside the axons. The area just below the axon hillock is called the initial segment of the axon, and this is the actual where the action potential is start. So this is the dendrites. Here the area of the pericardium with the nucleus, with the ribosomes, with rough endoplasmic, the nasal substance. This is a triangular area or the pyramidal shape which lack rough endoplasmic and ribosomes and myelin sheath, which is the axon hillock, and the area just below it is the initial segment of the axons. Again, this area here, the axon hillock, and you can see here this is the ribosomes, rough endoplasmic, prominent nucleolus, and the nucleus. As I said, in the axon we contain microtubules, and these very important in the transport of the metabolites inside the axons. There is two types of movement in the axons. We have the orthograde, which are from the cell body to the nerve ending, and retrograde, from the nerve ending to the cell body. So from the cell body toward the organ, this is the orthograde, whereas the retrograde, which is from the nerve ending toward the cell body. And this is very important because in the retrograde flow, some viruses, for example, the rabies and encephalitis and toxin, may be transmitted from the periphery of the body toward the cell body, toward the central nervous system. And for example, the rabies, which happened by bitten by a rabid dog, can cause death for a human and animal if it's reached to the central nervous system. Dendrites, which is branching of the cell processes, and usually it's protruded from the cell body. There is no myelin sheath. Very important thing, transmit the information toward the cell body. So the axon transmit the information away from the cell body, whereas the dendrite transmit the information toward the cell body. Large dendrite may have nasal substance, contain microtubules, but fewer neurofilaments. So all we can see here, like if there's one, this is the axon, and this one with a branching, this is the axons, and all of these branching structure here, all of these structure are the dendrites. Now we're going to explain the synapse. Any contact between two neurons, we call it synapse. And usually the synapse could be between the axons and dendrites, so we call it the exodendritic. From the axon with the cell body, axosomatic, axon with axon, axoaxonic, axon with dendritic spine, axospinous, dendrite with dendrite, dendrodendritic synapse. So we can see here any contact between two neurons that depend between the contact area between the two neurons, which part is contact the other one. So here we have the axosomatic, axon with the cell body, axon with dendrite, axodendritic. If we have this structure here, which is dendritic spine structure found in the dendrite, we call it axospinous synapse, dendrite with dendrite, dendrodendritics. And usually in the area of the synapse, we have the presynaptic element and we have the postsynaptic element. Here we have presynaptic area where we have this structure almost like light bulb. We call it presynaptic knob or button. And this area here, the space between the presynaptic and postsynaptic, and usually at this area here we have the neurotransmitter where will be released and then will be uptake by the postsynaptic elements, which could be dendrite or soma or axons. This is the area again. This is the area of pre-synaptic element, postsynaptic. This is the area of synaptic cleft, and here we have the neurotransmitter. As I said, they will be released and then uptake by the postsynaptic elements. Now I'm going to talk about the cells of the nervous system. I'm going to talk about the supporting cells, which are the neuroglial cells. And usually they found in the nervous system, in the gray matter and the white matter. And usually, as I said, there's no connective tissue in the nervous system as supportive tissue. Instead, we have cells. And these cells provide support, nourishing, and protection function of the neurons. And usually, as I said, they're found in the central nervous system, and they have also found in the peripheral nervous system. Central nervous system, which is include the brain, cerebrum, cerebellum, brainstem, and the spinal cord, whereas the peripheral nervous system, which is the nerve, spinal nerve, and the cranial nerve. In the nerves, in the peripheral nervous system, only we have one cell, which is the Schwann cell, whereas in the central nervous system, we have astrocyte, oligodendrocyte, microglial, and ependymal cells. And usually the glial cells are more numerous than neurons. For each neuron, we have around 10 to 50 cells that support these neurons. However, not all of the function of these neuroglial cells is completely understood. But if they are there, there should be have a function. So neuroglial cells, as I said, they provide mechanical and metabolic support of neurons. In the central nervous system, we have 4 cells: astrocyte, oligodendrocyte, microglial cells, and ependymal cells. Whereas in the peripheral nervous system, we only have one cell, which is the Schwann cell. Astrocyte is very important because it contributes to the formation of the blood-brain barrier, the barrier between the blood and the brain tissue, and very important because you don't want any toxic material or overdose from the minerals to reach the brain. So this is barrier control what goes in, what goes out from the brain tissue or to brain tissue from the blood. So the first cell is the astrocyte. It is the largest cell of the neuroglial cells. Their cytoplasmic processes branching, they have branching process or processes, and the distal portion almost like a feet and attached to the blood capillary, and these processes is called the vascular feet, and as I said, attached to the capillary wall and very important in assist and maintenance of the electrolyte balance in the central nervous system, and also provide a passageway for nutrient transport toward the neurons. The other cells we have is the oligodendrocyte. It's small angular cells. It's smaller than the astrocyte. Its embryonic origin is the neuroectodermal cell, the same as astrocyte. It's analogous to the Schwann cells that found in the peripheral nervous system, and this cell is very important in the formation of myelin sheath in the gray matter. The third cell we have is the microglial cells. This cell, the embryonic origin is different. This cell is derived from the bone marrow monocyte. So the origin of this cell, monocyte, where the other cells of the neuroglial cells, their origins in the neuroectodermal cells. As I said, these cells, they have very long cytoplasmic processes. The distal end is called the vascular feet, which attach to the blood capillary. The fourth cells we have is the ependymal cells, and this is very easy to find because these cells line the neural tube. So if we go to the neural tube and the ventricles of the brain, the lining of these cavities will be the ependymal cells. Usually ependymal cells with the blood capillaries responsible for production of the cerebrospinal fluid. And the name of the structure, which the ependymal cell with the blood vessels, we call it the choroid plexus. And as I said, it's very important because it's responsible for production of the cerebrospinal fluid. The only cell that found in the peripheral nervous system is the Schwann cell, and usually these cells wrap around the axons, and this will result in the formation of myelinated and non-myelinated nerve. The origin of this cell, very important, the origin is neural crest cells. So we have um, ependymal cells, neuroectoderm, oligodendrocyte, neuroectoderm, astrocyte, and neuroectoderm as embryonic origin. And the microglia is monocyte, whereas the Schwann cells, the origin are neural crest cells. How we could differentiate between them? If these cells in the same slide, the largest will be the astrocyte, the smallest is the microglial cells, and the middle size will be the oligodendrocyte. But this is very difficult to discern because they are different section, and you have to have the three in the same section to be able to see. This is the largest, the astrocyte, and the smallest is the microglial, and the middle size is the oligodendrocyte. We can do special stain, but here we have to use a special stain, and usually like we stain it for almost 2 or 3 months to get this structure, and this is all of these cells are the astrocyte. As I said, the easiest one to find these cells here, which is the ependymal cells, which line the neural tube and the ventricles. As I said, these cells with the blood vessels responsible for the formation of the cerebrospinal fluid, and these cells usually columnar cells but without a basement membrane, so these cells are epithelioid cells.